Okay, hi everyone. My name is Allison, and I'm here to talk about our advancements in identifying some T cell receptors and their associated antigens in a pigtail macaque model. So the goals and objectives of this study. Sorry, I'm tall. The goals and objectives of this project were to use a pigtail macaque. I'm really tall. Pigtail macaque model in order to identify Cocci-specific T cell antigens from a list of 27. And we're hoping to confirm previous T cell reactivity of vaccine protective antigens and compare those between pigtail macaques and humans in order to validate that model. We're also hoping to use the identified T cell receptor sequences in order to track valley fever protective responses moving forward in vaccination and challenge studies. So a little bit of T cell receptor background. These are composed of an alpha and a beta chain, which are composed of gene segments V, J, C, and D for the beta chain. And the recombination of all of these gene segments can theoretically generate up to 10 to the 15th unique T cell receptors. I looked it up, and that's a quadrillion different potential T cell receptors. And these basically act as barcodes that we can identify and track later. And of course, we all know that T helper cells are protective against Coccidioides, but however, not all subtypes of T helper cells are protective. So Th1 and Th17 subtypes are particularly helpful. Our questions here are which Coccidioides antigens were immunogens in natural exposure, and which T cell receptor clones and T cell phenotypes are associated with which of those antigens. And of course, moving forward, we would like to see are the same antigens reactive in previously exposed pigtail macaques and human patients. And why are we using pigtail macaques as a model for valley fever? Well, the pigtail macaques are susceptible to valley fever, and they have a similar disease incident rate as well as disease presentation to humans. And since we know T cells play an important response in vaccine-induced protection, we're also interested in looking at them in pigtail macaques. And the C segment is highly conserved on T cell receptors between humans and pigtail macaques. So in order to do this, we sampled PBMCs from pigtail macaques in both non-endemic and endemic areas. So we have Cocci-naive and naturally exposed pigtail macaques. We isolated their PBMCs and non-specifically expanded them for about 10 days. After this 10-day expansion, we stimulated them overnight with overlapping peptide pools of our Cocci antigens. We were then able to sort activated T cells and perform bulk T cell receptor sequencing on those cells. And this came from a list of 27 spherular expressed antigens. So we used this super cool multiplexing scheme, the MIRA method. MIRA stands for multiplex identification of T cell receptor antigen specificity. I guess MIRA sounded cooler than MITRUS, so they left out a couple letters in that. But we took each of our 27 antigens, as you guys see along the left, and we aliquoted our pigtail macaque cells into 7 aliquots, A through G. We then assigned each of our 27 antigens to a unique subset of 3 of these 7 aliquots. So for example, antigen 1 went into pools C, D, and E. Its address is CDE. No other antigen got that same address, but each antigen went into a subset of 3 of those 7 aliquots. We sorted out the activated T cells from each of these aliquots and sequenced them. When we got our sequences back, we can see if we see a T cell receptor sequence in, for example, pools C, E, and F, we can go and find that address and associate it back to the antigen that was in that unique address. And that is how we can associate T cell receptors with the antigens they were reacting to. And so what we saw here, we see on the y-axis, we have a TCR clonotype count, and along the x-axis is all 27 of our antigens. So in the naive animals at the top, we see only 3 of the 27 antigens associated with greater than 10 clones across our animals. And so this low number of T cell receptor clonotypes associated with all of the antigens in these naive animals is likely due to cross-reactivity from similar epitope exposure in the past. And we also see a pretty high level of background when we stimulate with this pool, because this includes over 1,000 peptides. And so when we look at a smaller subset, the NAU 108 that Marilene talked about earlier, and also go see Mom's poster number 43 in the poster session to hear more about this, we see a much lower level of background in those peptides. In our seropositive animals, these are animals that at some point have tested positive for Cocci. However, they had milder disease or have completely recovered from the disease. We see a much higher number of T-cell receptor clonotypes associated with these antigens. And to us, this is indicating potential clonal expansion in response to that pathogen. And then a memory response when we're stimulating with these 27 antigens. And importantly, we're seeing a couple of these antigens only in the seropositive animals and not in our completely naive group. Finally, we have this group of severely diseased animals. So these are animals that have disseminated Cocci, and many of them have succumbed to the disease. And we see a really low level of T-cell receptor clonotype counts across all 27 antigens. And of course, to us, this is telling us that these animals have not mounted a proper immune response to the disease, and so we're not seeing any circulating memory T cells that are then responding to our antigens of interest here. And so now that we know that we have some T cell receptors that are reacting with some of the antigens, we wanted to know more about those phenotypes of the cells that are reacting. And so we performed single cell sequencing. So far, we've only done this in one animal, one seropositive animal, but we did single cell sequencing in order to look at the phenotypes of the T cells. And so, as I mentioned, this is a seropositive animal that has controlled infection, and we were able to successfully single-cell sequence this pigtail macaque, which nobody has done before, and we were able to identify a whole bunch of T cells, which was very cool. So in total, we saw about 1,600 T cells. We were only able to associate about 308 of them with VDJs, but we were still able to get a lot of really cool information out of those 308 cells. So in these 308 cells, we see a couple of pretty big clusters forming. And so in this plot here, anytime you see two circles touching each other, those are two identical clones that we're seeing. And so we're seeing a couple of pretty big clusters, and we think this is associated with clonal expansion in this macaque. And importantly, these clusters, these cells that are clustering together, are only showing up in the sample that we stimulated with the 27 antigens. It's not showing up in our negative control. And so these are cells that were activated in response to the stimulus of the 27 antigens. Importantly, a whole bunch of them were also associated with a Th1 or Th17 response. And so we all know that the Th1 and Th17 are considered protective. And again, this animal was a seropositive animal that was able to control the infection. And so we think that moving forward, if we look at single cell sequencing in more of our seropositive animals, we may see a decreased Th1 and Th17 response in those animals. And so just to summarize what we found here, 24 of our 27 antigens of interest showed some level of activity in previously exposed pigtail macaques. 6 of these antigens are antigens that have been previously associated with protection against Cocci. And then we also saw 18 additional antigens in the study not previously associated with protection. And so moving forward, we need to perform more single cell sequencing in order to associate the T cell receptors with their antigens they're responding to and to see what are the phenotypes of those cells responding. Are they actually helpful protective responses, or are they detrimental? And so as I was saying, we're going to do confirmatory studies, more single cell sequencing, as well as ELI spots. Again, go see Mom's poster number 43 to hear more about ELI spots. And we're hoping to pair those T cell receptors to their phenotype to their reactive antigens. And we're also interested in potentially looking at paired bowel samples and PBMCs to see if there are specific T cell receptor clonotypes that are migrating to the primary site of infection. And of course, we're interested in examining the antigens of interest in humans to compare how similar the responses are in humans and pigtail macaques. And thank you to everyone who contributed to this project.
Hi, thanks. Great talk. Doug Lake, ASU. Are the pigtail macaques inbred or outbred?
They're
Outbred. Okay. And so are the peptides mapped to their MHCs, or and then the follow-up question? Well, I'll let you answer that question first.
So the peptides that we selected, we did map to MHCs to look at predicted binding affinity, and we selected only ones that had a greater than 15 percentile rank for predicted binding affinity.
Got it. So were there any antigens? So then you can know which peptides are most reactive that belong to particular antigens. Were there any that were head and shoulders above the others, or were they all similar of the antigens that you showed, like CTS1, PMP, et cetera?
We definitely had differing numbers of peptides for each antigen that passed that threshold of the 15 percentile rank in terms of binding. But since we only picked peptides that were above that threshold, then I guess we see similar predicted binding affinity across all 27.
No, the affinity is one thing, but what about the reactivity, the response of the T cells? Was PMP better than Eli antigen 1, for example?
We have not been able to look at that yet,
Okay.
but it's something we're interested in looking at moving forward.
So I was a little confused on one part. So you did TCR sequencing of each of those cells, and the ones that you highlighted were the ones that mapped to your expected epitopes based on your antigens. So, correct?
Yes.
Okay.
So, yeah.
So the rest of them that are unknown, are you going on then to look and try to define what those epitopes might be and kind of what the antigen recognition might be on those TCRs?
I think that's definitely an interesting future direction, but I think for now we're more looking to identify which of those antigens are that we know are eliciting the greatest response across the animals to try to narrow those antigens down for vaccine development.
Because you had quite a bit that weren't falling into those colored ones. So I'm just wondering if there was something else that might be a dominant epitope response that you're missing.
Yeah, potentially. And of course, we've only done the single cell sequencing in one animal so far. And so I think as we get a greater cohort going for that, we'll be able to glean a lot more information from that data set.
Wonderful. Well, great thesis work and congratulations on your defense yesterday.
Thank you.
I'm gonna just bring that up.
Any more questions? Okay, thank you, Allison. Now our third presentation is by Dina Harmon from Lawrence Livermore National Laboratory. His topic is single-cell transcriptomic analysis reveal unique patterns of myeloid cell differentiation and potential therapy targets during C. persistence infections.